Hi, I am Dr. Arsalan and today we are going to discuss the structure of skeletal muscles. The muscles which are attached to the bones or skeleton are called skeletal muscles. They are basically joined to the skeleton by connective tissues called tendons. There are more than 600 skeletal muscles present in the human body. These constitute 40% of the body weight. Skeletal muscles are red due to the presence of myoglobin, the oxygen storing pigment. So here we'll discuss the structure of skeletal muscles. The skeletal muscle layer is basically protected by a connective tissue layer called the fascia. Skeletal muscles are basically arranged in the muscle bundles. These muscle bundles are in turn protected by a layer of connective tissue called epimysium. And muscle bundles are of 2 cm length. Then in each muscle bundle, the muscle fibers are arranged in discrete groups. These groups are called fascicles. This fascicle is again shown here. The fascicles are covered by another connective tissue layer called perimysium. Perimysium is a connective tissue layer which protects the fascicle. The fascicle has diameter of 1 to 5 mm. And in each fascicle, this is called muscle fiber. And muscle fiber is protected by a layer of connective tissue called endomysium. Diameter of muscle fiber is 10 to 100 micrometer. This is the basic contractile unit of the muscle, so we can say that the muscle fibers are the cells of muscles. Fibers are also called as myocytes, are the cells of the muscles. Then in each muscle fiber there are myofibrils. Myofibrils are of 1 to 2 micrometer diameter, and in these myofibrils there are myofilaments. These myofilaments are of 8 to 16 nanometer width, and these can only be seen through electron microscope. Myofilaments are of two types, the actin filaments and the myosin filaments. So this was the basic structure of skeletal muscles. So if we consider their visibility, the muscle bundles and fascicles can be seen with the naked eye. The muscle fibers and myofibrils, these can be seen with the compound or light microscope, while the myofilaments can be seen with the electron microscope. These muscles are basically protected by the layer of connective tissue. For example, the muscles are protected by the fascia, the muscle bundles are protected by the epimysium, the fascicles are protected by the perimysium, the muscle fiber are protected by the endomysium. So these are the layer of connective tissue which surround and protect the skeletal muscles. The muscles are present in the form of muscle bundles, and in each muscle bundle 100 and 1000 of the muscle fibers are present in discrete groups. These groups are called fascicles, and from each fascicle the muscle fiber arises, the myocyte arises, giving rise to the myofibrils, and the myofibrils give rise to the myofilaments. Now here we will discuss the muscle fiber. This is particularly the muscle fiber, and it is the contractile unit of the muscles. Muscle fiber is the contractile unit of the muscles, and it is also called as myocyte. Muscle fibers are elongated, unbranched, multinucleated cells. Because of the huge length of the muscle fibers, many nuclei are present in the muscle. For instance, in some muscle cells there may be 3000 nuclei per cell. So thousands of mitochondria are also present in each muscle fibers. And it should be kept in mind that the nucleus present in the muscle fibers are peripheral. These are attached to the sides or walls of the muscle fibers, so they have peripheral nuclei. The cell membrane of the muscle fiber, it is called sarcolemma. The matrix present in this muscle fiber is called sarcoplasm. The endoplasmic reticulum present in these muscle fibers are called as sarcoplasmic reticulum. Skeletal muscles lack smooth endoplasmic reticulum. There is only rough endoplasmic reticulum which accumulates the calcium ions. Therefore, this is called as sarcoplasmic reticulum. And also the sarcolemma, the cell membrane of the muscle fiber, innervate deep into the cell transversely, forming a T-tubule. For example, this is the cell membrane. It innervates deeply into the muscle fiber. This is called T-tubule because it is innervating transversely. T-tubule means transverse tubule. And this is required for the transmission of run impulses across the muscle fibers. The length of the muscle fiber is 2 to 10 cm on average. It should be kept in mind that all the muscle cells starting from the muscle bundle to the fascicle to the muscle fiber are fibrils and myofilaments. All are running longitudinally in parallel fashion. Now we will discuss the myofilaments, the actin and myosin myofilaments in detail. If we consider this diagram as the thick and thin filaments, these are the thick filaments. Thick filaments are also called as myosin filaments because thick filaments are made up of myosin, and these are thin filaments. Thin filaments are made up of 3 proteins, the actin, the tropomyosin and troponin. These 3 proteins actually constitute the thin filaments. The thick filaments give dark region. These are called as dark bands. Dark band is also called as A band. A band means anisotropic. Anisotropic means that it can convert the light into polarized form, and this polarized light can travel in a plane. So this is also called as plane polarized light. Therefore, these are called A band, while the thin filaments, these are called I bands. I band means isotropic. Isotropic cannot convert the light into polarized form. These are also called as light bands. The thick filaments are of 16 nanometer diameter, while thin filaments are of 8 nanometer diameter. These are named thick and thin filaments due to differences in their diameter. Thin filaments are bisected by a line, a longitudinal line called Z line. Z means zewish. Zewish means middle. So this zewish line or Z line bisects the thin filaments at midline. Therefore, it is called Z line. So the distance between two Z lines, this is again another Z line. Distance between these two Z lines is called sarcomere. And sarcomere is the contractile unit of the muscles. In case of thick filaments, there are two regions. One region is being overlapped by the thin filaments, in which both thick and thin filaments are present. This is called overlapping region. While the central region, this is brighter end, it is called as bright region or light region, or we can say H zone. H means helle, and helle means bright. Thick filaments are present in this zone, and this zone, H zone, it is again bisected by M line. This is the M line, and this M line basically joins the two thick filaments. These two thick filaments are facing outwardly, these are oppositely facing from each other. This is the myosin head, and this is the myosin tail. So this is also called as tail to tail attachment of the myosin or thick filaments, which are attached to each other by the M line. So this is the electron micrograph of the skeletal muscles. There is thick filament and thin filament bands. These are called dark bands, and thin filament are called light bands. So as we have noted here that there are alternatively thin filaments, thick filaments. These give striped or striations to the muscle. Therefore, these skeletal muscles are also called as striated muscles because of alternative dark and light bands. These are called as striated muscles. During the process of contraction, the sarcomere shrinks because the myosin head crawls on the actin filament. This I band, or we can say the light band, or thin filaments are contracted. The size of sarcomeres is reduced during the contraction, and the thick filaments are constant in size, and these are never shrinked or never reduced. Only the thin filaments come close to each other, shrinking the size of sarcomeres, so muscle contraction occurs here. So during the muscle contraction, only I band or thin filaments contract. In dark band, about 300 myosin molecules join together M line. In dark band, basically their tails are joined at M line. We will discuss the thin filaments. Thin filaments are made up of actin proteins, the tropomyosin and troponin. Actin proteins are basically globular or spherical in shape, so these are called as G actin. And these are arranged in filamentous form, so we can also say that the actin are F actin. There are myosin binding sites on the actin proteins upon which the myosin head crawls. Myosin molecules basically crawls on the actin molecule, and in this way contraction takes place. So these binding sites are covered by the tropomyosin proteins. The tropomyosin proteins covers these binding sites on the actin proteins in case of relaxed or rest stage. And the troponin proteins, these accumulate calcium ions. When these absorb calcium ions during the nerve impulse transmission from the sarcoplasmic reticulum, these change their shape and thus hereby remove the tropomyosin from this binding site. And binding site is relieved, so myosin molecule again crawls upon this actin filament and contraction takes place. As we have seen in the muscle fiber, that the cell membrane of the muscle fiber was called as sarcolemma, which penetrated or innervated into the muscle fiber cell transversely, so formed T-tubule. Basically, during muscular contractions, these nerve impulses are transmitted from the central nervous system to the sarcolemma of these muscle fibers. And this sarcolemma communicates these signals to the T-tubule pass through the muscle fibers. And when these nerve impulses are received by the matrix in the sarcoplasm, the sarcoplasmic reticulum releases calcium ions. These calcium ions are accumulated by the troponin molecule, which change their shape and release the tropomyosin molecule from the myosin binding sites on the actin filament and crawl one to the next binding site. And in this way, muscle contraction takes place. If we consider this sarcoplasmic reticulum, so T-tubule innervated through these two sarcoplasmic reticulum, this formed a triad. So this is also called as muscular triad, in which there is a single T-tubule with adjacent sarcoplasmic reticulum. Now here we will discuss the polarized light. Seen that the 8 band, these were anisotropic, and these were capable of polarizing the light. was the capability of myosin molecules. For example, this is a myosin molecule or myosin protein. When the light was falling upon these molecules, this myosin molecule converts these light waves into a single plane. This was said x-axis or a plane by polarizing the light. These light waves or photons were polarized, forming the crust and trough upon this single plane. These crusts are positive charged and trough are negatively charged. So this light was basically polarized into positive and negative charges, and this light now travels in the single plane on x-axis. This is called polarized light. Prior to this myosin molecule, this light was non-polarized. These are the isotropic bands, while these are the anisotropic bands. So this is basically a difference between the polarized and non-polarized light, or anisotropic and isotropic band. So this was all about the structure of skeletal muscles. Thank you.